This is the first video lecture for Friday, October 26th. We're going to finish talking about the process by which cells make proteins based on information stored in DNA. Or in other words, gene expression. Taking a gene that's in DNA, expressing that gene, which means that it's used to do whatever the function of that gene is, which for a lot of genes is to make proteins. And we saw earlier that in order to do that, there's got to be some intermediate, which is RNA. RNA takes information from DNA and then takes that information to ribosomes, and that's the site of protein synthesis. So continuing on with this last day of these video lectures, you guys have been watching these videos, and this is the last couple. And so you're taking advantage of these videos, treating them just like a regular class, getting the information from here. For some people, it's a better situation than a regular class because you can stop these videos and make sure that you're caught up with your notes, or you can watch them again, whatever. But that's what we're doing. And all this material at the end of this week, we've covered a lot of material, is going to be it's going to be material used for some of the questions on the exam that we have on October 31st. So we've looked at RNA, what RNA does. It's this intermediate between DNA and proteins. We looked at the structure of RNA, saw that it's very similar to DNA. There are some differences, though, that are related to its function. We looked at these three different types of RNA, and you've familiarized yourself with those. So when you see messenger RNA, transfer RNA, and ribosomal RNA, then you know exactly what they are, and you're not having a hard time remembering the difference between those three different types of RNA. And then we started looking at the actual process of going from DNA to a protein. And in this case, if you're spending your time reviewing this topic, then you're not just making flashcards and saying, oh, what's this, that's that, what's RNA, messenger RNA, what does it do? Like a word on one side of a card and an answer on the back. But you're reviewing the whole process, how this actually works, because it's a process that takes place. It's not like the cell stops and says, okay, here I am in this, I'm done transcription, now I'm going to do translation. And that's where we are right now, is how you actually make proteins. So to remind you, we're looking at this whole process. We've gotten to the point where DNA was used to make RNA, messenger RNA. That was the process of transcription. And the language for both DNA and RNA is nucleotides, sugar, phosphate, base. They're not the same nucleotides because one is RNA and the other one is DNA, but yet they're both nucleotides. So it's a transcription process from DNA to RNA. And now we're at the point where this messenger RNA that's been synthesized based on information in DNA, so there's a gene that was used to make this strand of RNA, leaves the nucleus of the cell, goes out to the ribosome, and here we are. So this is where we are right now. You've got information in DNA, which is a chain of nucleotides. You've got information in RNA after it's finalized before it leaves the nucleus. That's a chain of nucleotides. Nucleotides aren't proteins. Nucleotides don't carry out all these different processes. So what we have yet to do is look at the translation of a language of nucleotides to a language of amino acids. A chain of amino acids, once it's modified, becomes a protein. And that moves things across the cell membrane. That moves things within an organism, muscles. That forms different parts of the cell structurally. It forms a whole bunch of different functions. those proteins. So that's where we are. How do you get from a chain of nucleotides in RNA to a chain of amino acids in proteins? Nucleotides to proteins. Nucleotides to proteins. That's the translation that's taking place here. Okay, so you've got messenger RNA that was made, messenger RNA that contains the information to make a protein. Messenger RNA has left the nucleus and shown up it's escorted to the ribosomes. So in these figures, you're going to see messenger RNA with these little green, looks like a comb or half of a railroad tracks. Each one of those little T parts of messenger RNA is a ribonucleotide. It's got a sugar and a phosphate and a base. And all of these ribonucleotides, the order that they're in, one base after another base after another base, is determined by the sequence of bases in DNA. So it's very specific. And this one little section of messenger RNA corresponds to one of thousands of genes in DNA. So this little green strand of messenger RNA is going to the ribosomes, and it's going to be used to make a particular protein. Not just any protein, but a protein that's coded for by the information in the gene that's in DNA. So here comes messenger RNA to the ribosome. And on this messenger RNA, there are 3 bases at a time that make up what's called a codon. It's 3 bases. And remember, the bases that are in messenger RNA are A and U and G and C. So 3 of those in a row, AUC, AUG, GCG. 3 of those make up a codon. It's actually not just the bases, but it's the bases and the sugars and the phosphates with each one of those. But sugars and phosphates all the same. It's the bases that differ. So we can concentrate on the bases. So it's when it's talking about, when it has these letters, A, U, G, it actually refers to the whole nucleotide, which is not just a base, but it's a sugar and a phosphate as well. But as I said, sugars and phosphates are all the same, so you don't even need to differentiate. That's what's on this messenger RNA. Codons, 1, 2, 3 bases, 1, 2, 3, 1, 2, 3, 1, 2, 3. Those are codons. What you find on the transfer RNA, this kind of T-shaped RNA here, 2 different parts, remember. If you don't remember this, you can go back and look at that other PowerPoint. But this transfer RNA has got an anticodon part with 3 bases. This is RNA. So those 3 bases are going to be one of 4 types. Those 3 bases here are, it's going to be an A, or it's going to be a U, or it's going to be a G, or it's going to be a C. That's what these 3 bases are going to be, one of those 4. A, U, G, 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 C, whatever it happens to be. The other part of this transfer RNA has a binding site, very specific binding site for one type of amino acid. There's 20 naturally occurring amino acids. So there's 20 types, at least, of transfer RNA. So this one, for instance, you probably can't read this, but this says MET. That is one particular amino acid, one of the 20 that there are. And that is methionine. So if you look at a transfer RNA molecule, it's got a methionine on it. And if it has a methionine, then it's only got one anticodon, one sequence of bases in that anticodon. It's going to be UAG. If this transfer RNA has a methionine attached to it, then its anticodon sequence is AUG. If this is not AUG, if it's something else like AAG or GGC, it's going to have a different amino acid attached to it. That's how this works, is that you've got a codon here in messenger RNA. These 3 bases or a certain sequence in a certain location along this messenger RNA. What those 3 bases are saying basically is that the amino acid that goes right here, nowhere else along here, but the amino acid that goes right here is methionine. So in order to get methionine right here to begin with, then the transfer RNA, the only transfer RNA that's going to match up with this codon, those 3 bases right there, the only bases that are going to match up with that are found on a transfer RNA that has the amino acid methionine. So there's a codon in the messenger RNA. There's an anticodon in the transfer RNA. Those 2 bases match up in their pairs, AU, GC, GC, or CG, UA. That's how you get from a very specific order of bases in the messenger RNA, and remember that was determined by the order of bases in DNA. That's how you get from a sequence of bases to a particular particular amino acid. And it takes 3 bases to get 1 amino acid. We'll see why that is in a minute. So let's look at this in more detail. Now, this is zoomed in a little more so you can see the letters here. Here's messenger RNA that's come from the nucleus. It's been put together based on information in DNA. This is at the ribosome. Here's this very first amino acid that's going to come in. As you can see now, easier, it says MET that's connected to this transfer RNA. This is the only transfer RNA out there that's going to match up with this codon. This is UAG. So the anticodon is AUC. If there's another transfer RNA that doesn't have this sequence of bases, UAG, it's not going to form a bond here. And there's no way that another amino acid is going to be brought in. The only thing that's going to go right here is a methionine. And the way to make sure that it's methionine and no other amino acid is to have these bases in the transfer RNA here match up with these bases in the messenger RNA. Or in other words, have the bases in the codon match up with the anticodon. That's how you can ensure that you get methionine and not one of the 19 other amino acids. So let's go one step further. This methionine. Okay, so you're bringing methionine in there. What does that mean? It doesn't mean anything really, except you're bringing in some amino acid. But remember, we're going to form a chain of amino acids, which is going to turn into a protein. So you had this one transfer RNA, this green thing, bring in methionine based on the anticodon matching up with the codon. Now you've got the next 3 bases. That's the next codon. It's got a certain order. And that order determines the next amino acid that's brought in. And in this case, if you looked closely enough, you'd see this says PRO. That's an amino acid called proline. So the instructions in this green messenger RNA, the message that's being delivered to the ribosomes, is that to make this protein, first you put a methionine in. Then you bring in a proline. And this is going to go on for another 160, 200 codons and anticodons where you're bringing in one amino acid at a time. It takes 3 bases in a codon and 3 bases in an anticodon to get to correspond to one of these 20 naturally occurring amino acids. So that's the information in this messenger RNA, is that if you're going to assemble this particular protein, the first amino acid is methionine, second amino acid is proline, The third amino acid is one of those 20, and the next one, and the next one, and the next one. And you need to join those together in a very specific order in order to get the protein that's going to carry out that protein's functions. All right, well, let's go one step further. Then you had this first transfer RNA bringing in the first amino acid, methionine, right there, based on the codon and the anticodon matching up. Complementary bases, not the same bases, but complementary. And you've got proline coming in. This anticodon is going to match up with that codon, and now you've got methionine first, proline second. Now this peptide bond is formed between the methionine, which was the first amino acid, and the proline, which is the second amino acid. There's a bond that's formed between them, two peptides, and as this continues to grow and you have 3 amino acids and 4 amino acids, 5 amino acids, this is called a polypeptide. Some of you have probably heard of polypeptides. Now look what this transfer RNA is doing. This ribosomal unit is moving along the messenger RNA. So it's gone past this one, brought in a methionine. It's onto this one, bringing in a proline. And now it's going to move on to this one. Whatever that next sequence of 3 bases is, there's going to be a corresponding transfer RNA with a corresponding amino acid. Now this transfer RNA that brought in methionine, it's gone, it comes off. It can go into the cell. Well, it's in the cell. It can go and form another bond with another methionine molecule. You get this methionine from your diet. We can make some amino acids, and other ones we have to get from our diet. The ones we get from our diet are called essential amino acids because we can't make them. I'm sure some of you have heard of essential amino acids. Okay, so now this transfer RNA is gone. It's going to go look for another methionine. It can bring another methionine in here later. You've got 2 amino acids that are joined together, and you're ready for the third one. So depending on what protein it is, there's going to be another amino acid. So here we are, a number of steps along the way. Here's the ribosomes, and this green part again, remember, is messenger RNA. And so this messenger RNA has been going through the ribosomes, or the ribosomes have been going along the messenger RNA, bringing in methionine first, and then proline and tyrosine and whatever these other ones are, phenylalanine and leucine. So there's 1, 2, 3, 4, 5 amino acids that have been joined together in a very specific sequence that corresponds to this particular protein, no other protein, just this protein. Could be an enzyme, it could be a structural protein, it could be part of hemoglobin. But that's the instructions for making this particular protein were in this messenger RNA, and it got there from DNA, from a gene in DNA. So now this ribosomal subunits are going along the messenger RNA, facilitating this assembly of amino acids in a very specific order. Now this is a very short amino acid chain or polypeptide. There's only 5 of them here, whereas a real amino acid chain or polypeptide that's going to go off and become a protein is probably going to have 100 or 200 or somewhere in between amino acids. So 100 times or 200 times one of these transfer RNAs has come in here, matched up with the codon, and brought a particular amino acid. Now you get to a certain point in this messenger RNA where there's a sequence of bases here in the codon, in the messenger RNA right here. There's a certain sequence of bases where there's no match on transfer RNA. There's no transfer RNA out there that will match up its anticodon with these bases, this base sequence in the codon. So since there's no transfer RNA that matches up here, then this whole process stops. This is a signal for this whole process of assembling amino acids in a chain to make a protein to stop. So that's called a stop codon. There's a sequence of bases with no match, transfer RNAs, stop. The whole process stops. And then this release factor comes in and breaks up everything. These ribosomal subunits can be used again. This messenger RNA here can be broken down and used again. These little ribonucleotides, one little T and another little T, this whole thing can be broken up and used again, assembled back in the nucleus of the cell, and used to make new messenger RNA. So here you've got a polypeptide. It's a protein with this amino end and a carboxyl end. It's a regular protein. This has only got 5, and like I said, a more realistic protein would have 200 amino acids, not just 5. But it would have a carboxyl end and an amino end, which is what makes it a protein. Okay, so then looking at this, as you're going along, this is what happens on a broader scale. You've got this green thing that's a messenger RNA, a strand of messenger RNA that was synthesized based on information in DNA. The ribosomal subunits come together and they move along this messenger RNA. And now what's happening is this brown spaghetti-like wiggly line, that's a protein. This protein's getting longer and longer as more and more amino acids are joined together. And then you get to the end, you get to a stop codon, and there's no more amino acids to be brought in because there's no more transfer RNAs that match up with the codon. And then this protein gets released along with the ribosomes and the messenger RNA, and now you've got this polypeptide chain. And that polypeptide chain can be modified. There's different levels of the structure of proteins. There's just the sequence of amino acids. Then there's bonds that can be formed in between the sequence, in between the amino acids. They can form bonds with one another. They can form bonds with sulfur and things like that. And they can also join up with other polypeptide chains, other proteins. So there's different levels of organization of proteins. Here's an example. Hemoglobin. Everybody knows about hemoglobin. So hemoglobin has 4 polypeptide chains, or 4 proteins, basically. There's a gene in our DNA that has information to make messenger RNA that can be used to assemble a chain of amino acids that makes up this light pink polypeptide chain. There's another gene that we have in our DNA that allows you to make this darker pink polypeptide chain. And there's another gene in our DNA to make this light colored one, and there's another gene, a 4th gene, that allows us to assemble a chain of amino acids that results in this polypeptide chain. So you can see here the numbers. 2 of these have 141 amino acids. So 141 times a transfer RNA has come in and brought one amino acid, starting with methionine, and then another one, another one, another one. 141 times until it's made this. And the other 2, that process has taken place 146 times. And then at the end of 141 or 146 transfer RNAs bringing in an amino acid, then you've got this polypeptide chain, and these join together to form this group of 4 proteins that make up hemoglobin. This is happening all the time. We make so many red blood cells, and we go through so much hemoglobin and so many red blood cells in our body that these proteins are being synthesized all the time. Red blood cells are being made all the time. Hemoglobin is being made all the time. It's very important for us, and carrying oxygen in our blood. Okay,